To start our exploration of the nervous system, we need to start out with an understanding of the structure of a typical nerve cell. Here we have a motor neuron. So this is going to be in the peripheral nervous system, and its job is going to be primarily to send signals from the brain, let's say, out to muscles and things like that. What we have here are dendrites, cell bodies, and axons. Dendrites are receptors located on the cell body. Sort of looks like wild hair sticking out there. And typically they're going to be involved in picking up signals from other nerves and then sending them to the cell body. If that nerve that reaches the cell body, or that nerve signal that reaches the cell body, is strong enough, it will be sent out the axon to the next nerve. So dendrites are receptors, cell body is where you process the signal, and then the axon is where that signal can be sent out to other nerves. What we notice here are the insulating cells, or the myelinating cells of the motor neurons, and in this case they are Schwann cells. In the central nervous system we saw oligodendrocytes, and a difference here between the peripheral and central nervous systems is the ability to regenerate. And we'll get to that in a later slide here in terms of cell repair, but peripheral system nerves are capable of repairing themselves because of the encouraging effects, or the stimulatory effects, that Schwann cells have on nerve growth. In the central nervous system, if something is damaged there, the oligodendrocytes actually inhibit cell growth and repair. So we generally say that the central nervous system is incapable of repairing itself, and the peripheral nervous system is capable of repairing itself. The more insulation you have, the faster and more efficiently nerve signals can travel. Here we see Schwann cells wrapping around an axon, and it actually wraps around multiple times to have multiple layers of insulation there. And that helps to provide a greater speed of information transfer and a reduced likelihood of that nerve signal trying to go the wrong place. It also makes sure that the nerve is tightly wrapped in insulation. In this case, the insulation is going to carry out the same function that the insulation on the wires of your house carries out. And that is to make sure that electricity stays in the wire and doesn't try to jump out and go other places in the wall it's not supposed to go. It also helps that electricity, because it is staying more focused in the wire, to travel faster and farther. So in general, we would say that the more heavily insulated a nerve is, the faster it is capable of sending signals. If we look at different types of neurons based on their structure here, we can find multipolar neurons, which have dendrites attached to a cell body, which then has an axon coming out the other end. Bipolar neurons have a single dendrite, which may have multiple receptor antennae, attached to a cell body, which then projects an axon out the other end. A unipolar nerve has no dendrites, but rather has axon with receptor endings on one end and nerve signal transmitting receptor projections on the other end. And the cell body is sort of dangling off to the side on a little stem. If we look at some different examples of these types found throughout the body, the multipolar we would find in places like the cerebellum. And here we see the Purkinje cell of the cerebellum. And the cerebellum's job is to coordinate skeletal muscle function. Let's say the brain decides that you want to make a fist. The cerebellum will then determine how to execute that desire and which cells need to be used. And so this needs to be a very receptive type of cell. And we see here that the dendrite array for that particular cell is very large. Lots of dendrites, lots of receptors, so it should be a very sensitive cell. If we look at the pyramidal cell next to it here, it has a fair number of dendrites, but nowhere near the complexity that the Purkinje cell had, so we would expect it to be far less sensitive. In the bipolar category, we have olfactory cells and retinal cells. So we're talking here about smell versus vision. And what we see in both of them is a relatively small dendrite array. One dendrite, relatively small number of receptors. But if we're looking at the two of them, it looks like the retinal cell actually has more receptors than the olfactory cell does, which suggests that your vision would be capable of distinguishing a greater number of things than your smell can. So your vision should be better than your smell. If we were to look at some animals that rely more on one thing versus another, we might see a difference here. So we are visual, and yes, we can smell, but that's a relatively undeveloped ability that we have. If we look at a dog is capable of smelling thousands of times more than we are, so we would expect its olfactory cells to be much more developed and have a lot more of them than we would. And then their vision cells, well, dogs like to be able to see, but smell is more important. So you would probably see not necessarily much greater visual ability than we have, and perhaps a little bit less. A unipolar cell, in this case a dorsal root ganglion that we'll talk about in chapter 12 when we get to the spinal cord. It has, again, the cell body off to the side, receptive endings on one end of the axon, and sending signals endings on the other end. Here's just a quick look at some of those things. So the multipolar, sending signals to the muscles, we talked about that. The vision and eyes. And in the case of the bipolar and the unipolar, they're more of a plug-in. So they connect one thing to the next, and they're not necessarily so much involved in reception to some degree. So you might see them again being a little bit simpler of structure. These images should look familiar to you. We would have seen things that looked a lot like this when we talked about muscles. And you would recall in muscles, a neurotransmitter would be released from a nerve, and it would then bind to the membrane of a muscle cell. And so that's what we would see here in the chemically gated channel, is a neurotransmitter attaches, and then that causes, in this case, sodium and potassium to trade places. Sodium in, potassium out. In a voltage-gated channel, it's going to be run by electricity. So this would have been what happened when an action potential was generated. In the world of a nerve, an action potential can also be called a nerve impulse. Basically, that is, instead of creating a current to stimulate muscle contraction, we're creating a current to send a signal on out past that particular nerve. Both of these are involved in the process. We would see more of the chemically gated channels in the dendrites and cell body. We would see more on the line of voltage-gated channel in the axons themselves. So here's that process happening. We've got, in this case, a voltage channel here, and it's been stimulated. We've got sodium going one way, potassium going the other, and creating a change in electrical current. That change, creating the action potential, or the continuation of signal, as we've mentioned with muscles, needs to reach a voltage of about 30 millivolts. And that would be then enough to achieve, in this case, a nerve signal firing. Just like in muscles as well, there's a minimum. There's a threshold that you have to meet. So here we have this threshold voltage. Let's say it's the 30 millivolts. And our voltage was less than that. So we got no nerve firing. Down here, our stimulus exceeded 30 millivolts by some amount, and we got a certain number of nerve signals being fired. Just like with muscles, the stronger the stimulus, the more outcome you get. So here we had a much stronger stimulus than we had just to get started, and so we get a faster and more frequent nerve firing with a greater stimulus. What we've looked at so far is mostly the axon being insulated. But what happens in the dendrite? What happens in the cell body? And what happens in the case of axons that are not insulated? There we have this idea of a loss of signal. So the less insulated the nerve is, the faster the voltage decays, or it loses that signal. So let's say right here, we got our stimulus. So that signal came in, and it generated the firing of the nerve, and it sent that voltage out. But as it travels down this nerve, so in this case it's headed down the dendrite towards the cell body, we're losing voltage. The same thing would happen in your house if the electric wires weren't insulated. They would have less voltage at the end than they would have when it started. And even with insulation, there will be some decay of voltage. But if there's no insulation there, the decay of voltage will be very, very rapid. So if we look at that idea, if we want the signal to go a long ways, that uninsulated, so that graded potential is when you start out with a certain voltage and it very quickly decays, so your potential becomes less as you go. That graded potential only goes so far. And then the signal is so weak that even if it does reach the body of the nerve, it probably wouldn't send the signal on from there. So if you want the signal to go a long ways, and you don't have a lot of insulation, you have to have recharge stations. So here's this voltage gated channel here. We had our stimulus. So the voltage came along and stimulated that signal. It travels and it starts to decay. But when it reaches a certain level of decay, we have another voltage gated channel, which recharges the signal. It starts to decay, recharge, decay, recharge, decay, over and over. So you can then go a long distance. So this would be an uninsulated axon. You can go a long distance without having a lot of insulation, but you have to recharge often. That means it's slow, because you have to stop and recharge and stop and recharge. Sort of the idea of a purely battery-powered car. The battery's only good for so long, then you have to stop and recharge. That takes time. In myelinated axons, so insulated axons, because the insulation reduces the rate at which the voltage decays, you can travel a much farther distance before you have to stop and recharge. This allows for a much higher rate of signal transmission. And so this process is called saltatory conduction. What that means is the signal starts here, it's insulated so it goes a long ways before it gets weak, recharge at this gap between insulation, and then you can go to the next gap. And it appears as if the voltage is jumping from one gap to the next. Now it's not really, but the recharging effect only has to happen in between. So a recharge seems to jump from one gap to the next. And again, fewer stops to recharge, the signal gets there faster. Here we're seeing synapses. So here's one synapse right here. Here's another one right here. And basically a synapse is a connection between the axon terminals of one neuron. So here's the axon terminals that will release the neurotransmitter to the next one. And then it's going to be picked up by receptors here. So the axon terminals of one nerve connecting to the next nerve. Either here is the body, it's directly plugging into the body, or it could plug into a dendrite. Either way, a synapse is a connection between nerves. We can have a couple different kinds of synapses based on where the nerves are connecting. Axosomatic is our first one. The axon of the nerve synapses with the somite, or the cell body, of the next nerve. An axodendritic has the synapse, or the connection between the axon of one nerve and the dendrite of the next nerve in line. This is probably the most common one right here. And then we have an axoaxonal down here, where the axon of one nerve directly synapses with the axon of the next nerve, skipping the dendrites and the cell body altogether. So take a moment and think about these three different types of synapses. Which would provide the fastest nerve signal transmission? Remember, axodendritic has to go through the dendrite, through the cell body, then through the axon and out. An axosomatic would skip the dendrite going straight to the cell body, then axon and on out. But the axonal is the fastest of all three because it skips the dendrite, skips the cell body, and goes straight to the axon and on out. The more middlemen you cut out of the equation, the faster the signal will travel. When you talk about someone's brain developing and developing synapses, what you're talking about is brain cells connecting with other brain cells. So the more synapses there are in the brain, the greater the communication ability between brain cells. Therefore, the greater the processing capacity of those brain cells. So the more interconnected they are, the greater the processing capacity. So when you're encouraging learning and brain development, really what you're encouraging is the development of more synapses. So here's a close-up look at a synapse. This one happens to be between the axon of one nerve and the, looks like maybe the cell body or maybe a dendrite, it's hard to tell right here, of the next nerve. And so we have the neurotransmitter is released from the axon here. It's absorbed by these chemically gated channels here, which will then stimulate those sodium and potassium exchanges. And then once that's absorbed to a certain degree, it's going to go ahead and send the voltage-gated channel some voltage to work with and then stimulate further transmission of that signal. If we were back at the muscles, the receptors of the muscles that picked up the neurotransmitter would have been chemically gated channels. And then going down the membrane of the muscle would have been the voltage-gated channels. It turns out much of the body is all built on the same basic plans and principles, so if you understood the muscles, hopefully you're understanding nerve function a little bit easier. So I've mentioned graded potentials and I've mentioned action potentials. So the graded potentials were those that didn't recharge. They got a signal, they weren't typically insulated, and so the current, the voltage, very quickly decayed. And if you tried to send a message via a graded potential a long distance, it wouldn't work. So we're going to say graded potentials typically occur in the dendrites and in the cell body, where there's not that far for the signal to go anyway. So it's limited to short distances. Action potentials, because they have those recharge stations, and because there's often a deal of insulation associated with that particular part of the nerve, then this allows for long distance transmission. So again, graded potentials, short distance transmission, action potentials, long distance transmission. There are a number of different neurotransmitters that are utilized throughout the body for different kinds of communication. And we've seen acetylcholine already. And it's probably one of the most common because it is involved in muscle contraction, and muscle contraction does occur throughout the body, so it's no surprise we see it everywhere. Some others, and we're not going to go into all of these, but norepinephrine, that's adrenaline, and so that's going to help you feel good, and that would have a stimulatory effect on some parts of the body, and it would have an inhibitory effect on other parts of the body. When we get into later chapters, looking at chapter 14 specifically, we'll see some of those effects, where in one part of the body it stimulates it, in other parts of the body it inhibits it at the same time. And it will make sense, hopefully, when we get there. I don't know that it's possible to overemphasize dopamine, but dopamine is so incredibly involved throughout the body. It's involved in your mood and how you feel. It's involved in muscle contraction. It's involved in really every part of brain function. And so if there are problems with dopamine, either an inhibitory or a stimulatory effect is supposed to have, if the levels aren't right, there are serious problems. Histamine is involved in inflammation and swelling to some degree, as well as your level of awakeness and appetite and a number of other things as well. So these things are often multi-purpose. Not really going to spend a lot of time here, but one to mention is glutamate, and that seems to be significant in the problems that occur with a stroke. Endorphins are natural painkillers, so that's a pretty handy thing if your body happens to produce it sometimes. When we get down here to the purines and the gases and lipids, I start to have some issues with calling some of these neurotransmitters. If we're saying a neurotransmitter is a communicator between cells, then ATP, or energy, isn't necessarily a communicator. So I really feel that ATP is not properly labeled as a neurotransmitter. And when we get down here to the gases, nitric oxide is a stimulator of blood vessel dilation, and again, as a gas, having an effect on the body, necessarily a neurotransmitter. I'm not really sure about that one. Carbon monoxide is going to prevent transmission of signals in the brain to a great degree, and if the levels get too high, will kill you. So I don't know that we would call that a normal communicator either. But the one I have the greatest issue with are the endocannabinoids. And if you look over in the comments section, these are the receptors where marijuana seems to have an effect. So basically saying that these are neurotransmitters then says that marijuana is a neurotransmitter and that we are biologically programmed to expect to use marijuana. If we have neurotransmitters, or if it functions as a neurotransmitter, we have neurotransmitter receptors for it, then we should be programmed to use it. Now, some of you are perhaps going to like that idea, and you know who you are. I personally take issue with this idea, though, and I would say that yes, marijuana does impact nerve signaling, but should not be considered a normal neurotransmitter in the process. So I would suggest that's misnamed a little bit and is given some degree of functionality and significance to the body that it probably shouldn't have. Let's talk about reflexes. You know what a reflex is. If you put your hand on a hot stove, you immediately jerk it off. If you step on a nail, you immediately shift to the other foot. If someone jumps out and yells boo at you, you instinctively jump, throw your arms in the air, close your eyes, and pull your head out of the way. So all of those things are reflexes. You have no conscious control over them. But they do follow a very specific pathway. A 5-step process, if you will. And the first step is the receptor. So the stimulus is detected. Whether it's sound, temperature, whatever it happens to be. That then connects to a sensory neuron. That neuron leads to the spinal cord, which in reflexes, the spinal cord is the integration center. It's the processing center. So it integrates in the spinal cord, then the signal comes out on a motor neuron to an effector, which gets a response. In this case, the movement of the head, the closing of the eyes, the jerking of the hand, whatever that happens to be. But the spinal cord makes the decision in reflexes. Now, eventually it will tell the brain that, oh, by the way, this happened and I did this, but it doesn't consult the brain when trying to determine what to do. This is because reflexes are designed for one simple function: to avoid or minimize damage if possible to a potentially harmful experience. So think about the hot stove, the nail in the foot, the being yelled at boo. All of those things are potentially damaging, harmful, deadly to the body. So if you take too long to respond by sending the signal up the spinal cord, through the brain, back out the brain, back down the spinal cord, and finally getting something done, that might take a second or two. And in this case, a second or two you do not have. So the spinal cord immediately makes the decision, gets the job done, and then later informs the brain as a informational statement rather than a what should I do from here. It allows the whole process to occur much, much faster. We can have different kinds of circuits. So in your house you have different electrical circuits, and one circuit might have a certain number of plug-ins, another circuit might have a different number based on what you're planning on. You don't want to put all of the appliances in your kitchen on the same circuit. That would overload it and it wouldn't be appropriate wiring. So different functions in the brain have different, and throughout the body really, have different kinds of circuits as well. Our first example is a diverging circuit. So in a diverging circuit we have a single neuron that sends out a signal. And in this case it happens to branch out to two different neurons. Each of those branch out to two, and each of those branch out to two. So that by the time you get from, let's say, the brain where we said I want this thing to happen, and then the time we get out to the muscles where we actually make it happen, one signal from one nerve has diverged and perhaps activated hundreds or thousands of skeletal muscles by the time it gets there. So this is nice because, again, one signal activates multiple outcomes by the time the signal gets there. So another name for a diverging circuit is an amplifying circuit. So just like the name suggests, it takes something and makes it much bigger. So if you wanted to activate, let's say, 100,000 skeletal muscle cells, you wouldn't have to send 100,000 signals. You could send just a couple, perhaps, and by the time it diverged and diverged on its way out and got there, thousands would be engaged in the process. So that's a very efficient concept. A converging circuit is a concentrating circuit. And what this means is you can have multiple different inputs that all result in one particular output. So here we could have had input 1 could have triggered a particular output. Input 2 could have also triggered that same output, and input 3 could have triggered that same output. The more of these potential inputs are involved, the stronger the output. This is highly involved in memory. If we were in the classroom, I would consider sharing with you some of my more powerful memories. I'm not going to do that because it's actually quite difficult for me to share some of those because of this converging circuit concept. So think about the strongest particular memory that comes to mind for you, or the most dramatic memory. And did it have any of the following connections? Did it have a visual component? Did it have a sound component? Did it have a smell component? Could it have had a taste component? Odds are your stronger memories have multiple different components to them, so each one of those components would be an input. And any one of them, if you were to experience that particular piece, could trigger that particular memory as an output. If multiple of those stimuli are present at the same time, you are much more likely to get that output. PTSD with military experience seems to be a part of this process. So with that particular traumatic event that you would have experienced, there were a set of inputs. There were a set of things that were associated with it. And if you experience those things again, it can flash you back or put you back into that experience where you had that original traumatic experience. And the more of those criteria are together at once, the more powerful your response would be. A reverberating circuit is really neat in that it allows for repetition of signal with only one input. So let's say we had our one input here. It went to the next nerve, to the next, to the next, and got an output of some sort. As that nerve signal was traveling through, it would come out here and bounce back around, or reverberate, basically echo, and come back into the system. It could echo through here. It could echo through here. And any number of these combinations of circuits, it could circle around and around. And every time it came back into the circuit, it would send that signal out. So let's say we had one signal that came through here. We got one output, two outputs, three outputs, four outputs, 5, 6, 7, and each of those could have circled back around again. So we got a minimum of 7 different outputs from one input because it kept circling around and around in the system. And that could circle around perhaps indefinitely. And a nice thing about that is it allows for rhythmic, repetitive activity without repeated signaling. So let's think breathing. Let's think walking. Let's think waking up and going to sleep. All of those things are rhythmic, repetitive processes. So if you want to breathe, and you want to breathe 100 times, you don't have to send out 100 signals. Perhaps you send out 5 or 6. They go through these reverberations, and by the time the signal is worn out, it's actually provided those hundreds of outputs. So this allows for a reduction in signal frequency to give us regular outputs. Now, if you don't want a regular, rhythmic, predictable output, this might not work for you. But it definitely allows us to breathe without having to think about breathing. It would probably allow the heart to beat without having to think about beating, because the circuit keeps reminding itself over and over with minimal additional input. Parallel after discharge circuits allow for complex processing of information. So critical thinking would certainly be done in a parallel after discharge circuit. So in this case, we have our input. It can come straight through for an output, but it's similar to a reverberating circuit in that some of that goes out to the side. In this case, came out through 1, 2, down to cell 3. So this first signal would have gone through, it came through first. Then this one over here, diverging by 1 cell, would have come in. And then this one with 2 cells and this one with 2 cells would have came in. So we would have that information coming in at different points. And what this allows you to do is do complicated thinking. So let's take a math problem, for example. Perhaps you can do a math problem in your head. 2 times 2 is 4. 4 times 4 is 16. And you've learned that simply by repetition and memory. But when you get to the point of 16 times 16, that becomes a little bit different story. That requires you to take that problem, and some of you perhaps could do that in your head, but it would probably take a few minutes. And what you would do is you would disassemble the problem. You would take part of the problem over here and work it over here on this side. You'd work part of the problem here, you'd work part of the problem over here, and at the end you would take all those pieces to the problem, put it back together in your output, in this case your answer, and that's how you work the problem. I will give you another video in this section here that shows an individual who is capable of going above and beyond with parallel after discharge circuits. And what he does is he does math problems in his head. The sort of problems that might take some of you minutes, if not longer, to work out on a calculator, if the calculator could do it at all, but he's able to do it in his head because he can take the problems, break them into different pieces, work each piece by itself, and then plug those individual pieces back in to form the final answer. And so he does that with a number of different examples, some of which is squaring numbers that I just talked about here, the 2 times 2, the 4 times 4, 16 times 16. And then he does it with calendar dates, so works the math in his head on calendar dates and spits out day of the week. That's pretty impressive. And then he factors numbers in his head that sometimes a calculator can't do. So make sure you check out that other video. It's really good stuff. If you happen to be watching this video and you found it just out on YouTube and not from within my class shell, it is Arthur Benjamin, Faster Than a Calculator. So if you go on YouTube and look for Faster Than a Calculator with Arthur Benjamin, you'll find this video. It's about 15 minutes, and it's a fantastic example of parallel after discharge circuit function. Hopefully you learned a lot in this video, and it was informative and clear. And as always, have a good day. We'll see you next time.